Fleas can infect both wild and domesticated animals. They don't have wings, but have long, powerful legs which allow them to jump. They are 2 to 4 millimeters long and are about the size of head lice. However, they are compressed laterally, so they have a very narrow body when viewed from above. Both male and females bite and prefer specific hosts. For example, dog fleas prefer to live and feed on dogs, but if there are no dogs around, they will look for other animals such as humans to feed on instead. Fleas undergo complete metamorphosis, so they have larvae and pupae stages. They lay their eggs in batches on the host, but because animals move around and scratch, these eggs usually drop off onto carpets and rugs before they hatch. Once they hatch, larvae move to more shaded areas, usually under furniture, and become pupae after roughly a week or two. The cocoons of flea pupae are encrusted with debris, so they are hard to find because they look like particles of dust. After a few weeks or months, depending on many factors such as temperature or carbon dioxide levels, the adult fleas emerge. This is quite a long while, and if Fido has been sent to the farm during that time, the humans that remain will be the source of their first blood meal. Fleas can transmit a number of human diseases from infested animals. One of the ones you've heard about is plague. There is also another type of typhus that fleas can transmit called endemic flea-borne typhus. This is different to epidemic louse-borne typhus that we covered last week, and I'll talk more about this different type of typhus later on today. Fleas can also be intermediate hosts for the dwarf tapeworm, which can infect young children. However, the most common health problem related to fleas is an allergic reaction to flea bites, with small red itchy spots that are either clustered or linear, and there may be many more bites from fleas than from bed bugs. The most common fleas in homes are dog and cat fleas, which peak in the fall. Dogs and cats are the primary host, and you can see bald patches or hot spots on infested pets. As I mentioned before, these fleas will bite humans if pets are absent. Dog and cat fleas can be distinguished, but some experience is needed as they look quite similar, with spiny combs, and variations can be tiny, such as where the bristles are on the head or the shape of the head. Let's look at the oriental rat flea because this is the vector for plague. They are mainly found on wild rodents such as prairie dogs, and not such a great concern in urban areas in Ontario. More of a problem in the prairies such as in Saskatchewan, and in 2010 a plague-infected prairie dog was identified in a national park. Southwestern USA is more of a hotspot because of a large number of prairie dogs there. Rat fleas are easier to distinguish from dog or cat fleas because they don't have spiny combs, the mustache-like structures at the front and around their necks. There is also a type of flea called the human flea. Aside from humans, they can also infest pigs and other mammals, and are thought to have originated from South America. They have been reported on the Pacific coast and cause dermatitis and allergies. They can actually transmit plague, and this has been reported in the South American Andes. They look more like rat fleas, with no spiny combs, and it's harder to distinguish them from rat fleas than from dog or cat fleas. Diagnosis is by observation of fleas being present, and also bites that are usually on the legs below the knees for dog or cat fleas, and all over the body for human fleas. To help suppress itching, antihistamines and anti-itch creams can be used. Fleas can be controlled by sanitation. Vacuum cleaning will kill larvae, and steam cleaning is even more effective, killing all the stages. Washing bedding using the hot wash cycle will help to eliminate the infestation, and also other pests such as bedbugs and lice if they are present. Chemical control is also similar to other insect pests. Pesticide-grade diatomaceous earth can be used to dry them out, or methoprin, which is a juvenile growth regulator that targets the larval stage. To control fleas on animals, monitor them by inspecting them regularly, and use oral or topical applications if fleas are present. I've listed a few on the slide. Some recommendations are: don't wash your dog, before applying the topical spot-on formulations, as they need the skin oils to work properly. Make sure that formulations for dogs are not used for cats, because some can be toxic to different types of animals. For example, the pyrethroid permethrin is fine for dogs, but neurotoxic to cats. Over-the-counter flea collars, combs, shampoos, and dust that can be purchased from pet stores may not be as effective as those prescribed by veterinarians. The disease carried by fleas, plague, has an incubation period that can vary between 2 to 7 days. Initial symptoms may be flu-like, but then it may develop into different forms of plague. The most common form is bubonic plague. Lymph glands near the flea bite, usually in the groin area if the bites are on the leg, will become swollen, and they may even rupture. These swollen lymph glands are called buboes, and you can see a couple on the inner thighs of the person in the picture. More than half the cases will die if untreated. Another form of plague is septicemic plague, which is usually a progression from bubonic plague when the bacteria spreads to the bloodstream. Untreated cases die from endotoxic shock. The last form of plague is pneumonic plague, which is found in a small percentage of cases. This form of plague occurs when the lungs become infected, resulting in a cough with mucoid and bloody sputum. It is the most contagious form of plague and can be transmitted directly from person to person. It can also kill within days, and in many cases, parts of the body start to turn bluish-black, hence the term Black Death. This diagram shows different transmission cycles for plague. The most predominant cycle that we have today in North America is the sylvatic cycle, in which fleas become infected after feeding on an infected wild rodent. Humans become infected if they come into direct contact with either the infected animal or are bitten by the infected flea. The urban cycle is the one that occurred during the plague outbreaks in the Middle Ages, where domestic rodents were the main reservoirs. Humans became infected when they were exposed to infected rodents or bitten by infected fleas in their own homes. Pneumonic plague can result from either secondary spread from bubonic and septicemic plague, or through direct contact with another human or feline cases of pneumonic plague. So don't let any cats cough in your face, just in case. So to summarize, infected rodent fleas carry the plague bacteria Yersinia pestis. Bubonic plague is the main form of plague reported in North America, and is usually transmitted to humans from wild rodents through flea bites. Bubonic plague is not normally transmitted from one person to another. On the other hand, pneumonic plague is usually transmitted directly from human to human. The primary reservoirs of plague are wild rodents, such as prairie dogs. Other wild animals, such as coyotes, are incidental hosts when they are exposed to wild rodents. Cats can also become infected with plague, especially when they catch and kill wild rodents, and as I mentioned before, can transmit pneumonic plague directly to humans. This includes both feral cats, which are cats that were once domesticated but have become wild after a few generations, and pet cats. Around the world, plague persists in large areas, especially where there are infected wild rodents. In North America, it is reported mainly in the western United States, in the area surrounded by the 4 states of Arizona, New Mexico, Colorado, and Utah, and this area is known as the Four Corners. The last reported human case of plague in Canada was in 1939. However, it is still a national notifiable disease today because it is in the B list of biological warfare agents. Therefore, all forms of plague are reportable. However, only pneumonic plague cases need to be reported to the WHO. For laboratory diagnosis, different clinical specimens need to be submitted for different types of plague. For bubonic plague, aspirates from buboes are required, whereas blood specimens are required for septicemic plague and bronchial washings for pneumonic plague. Yersinia pestis is a gram-negative bacteria that belongs to the Enterobacteriaceae family, which includes gastrointestinal bacteria such as Salmonella, Shigella, and E. coli. When it's stained up, it has a bipolar staining pattern with more intense staining at both ends with a safety pin appearance. Immunofluorescence assays can also be used with antibodies to one of its antigens. Plague can be treated with antibiotics, and if diagnosed and treated in time, death can be prevented in more than 90% of cases of bubonic plague. To prevent and control plague, rodents and fleas need to be controlled. This can be achieved by rat-proofing houses and storing garbage appropriately. Insecticides and repellents can be used for fleas. In plague-endemic areas, pets should be treated for fleas. Wildlife and domesticated animals should be monitored for plague, especially rodents and cats. There are plague vaccines, but they only protect against bubonic plague and are not available in North America. Close contact of pneumonic plague cases should take antibiotics, as well as family members who share the same household with cases of bubonic plague. Families with human flea infestations should also be treated with insecticides, and hunters and veterinarians should use PPE, including gloves and masks, when handling infected cats. Endemic flea-borne typhus is also known as murine typhus. The bacteria that causes this disease is a different Rickettsia species to the one that causes epidemic louse-borne typhus. The symptoms are milder than louse-borne typhus, with lower case fatality ratios. Rashes may be present in half the cases, mainly on the palms of hands and soles of feet. Transmission is from flea-infested cats, dogs, or rodents, and not from human to human. Once again, it is not transmitted through a flea bite, but from contamination of the flea bite, or skin abrasions, or inhalation of flea excrement or crushed fleas. Endemic flea-borne typhus is found all over the world, especially in tropical and subtropical areas, mainly in ports and coastal regions that are infested by rodents. In North America, endemic areas include the Texas Gulf Coast, Southern California, and Hawaii. To prevent the disease, fleas should be controlled with insecticides in areas with rat infestations, and rodent control should be set in place. The provincial public health labs can identify various insect pests if they are submitted. Here are some figures of insect pests that have been identified after submission to the provincial public health lab. Although the data is old, it reflects the frequency of various pests in Ontario, with bedbugs and head or body lice being the most commonly identified.